Hi, welcome to what I hope will be the last screencast of the semester. We're going to do kind of the first half to two-thirds of the blood. I'll try and keep it brief. We'll see how far we get. So let's just dive right in. The blood, basically, if you think about what the bloodstream does, is it distributes everything everywhere. So oxygen from the lungs gets carried everywhere. Nutrients. When I say nutrients, you might think of something like glucose, amino acids, anything carbohydrates that you've ingested. Metabolic waste, you might be thinking CO2 might be going to the lungs, that sort of thing. And hormones we talked about in chapter 15, they get dumped into the bloodstream and go everywhere. If you recall from chapter 6, I think, whichever chapter the skin was, the skin works with your cardiovascular system to help regulate heat. So if you are overheating, more blood goes to the surface and you radiate out the heat. If you are cold, then those surface blood vessels constrict and you keep your blood and your warmth closer to your core. The blood also has chemicals in it called buffers that help maintain pH, and we have a whole chapter about pH and water balance. Your bloodstream also helps protect you, one, against blood loss. So there are proteins and what are called platelets, kind of like a cell, inside your bloodstream that cause clotting and coagulation when you spring a leak, and then that plugs the leak, which is awfully nice. Your bloodstream also protects you from infection. You have a number of different types of proteins or chemicals floating around in your bloodstream that have antimicrobial or antiviral properties to them. And then you have a host of white blood cells that produce some of these proteins and do a lot to protect you from invasion. We'll talk about all of that in chapter 20, I believe, is the immune system, or 21. Blood, if you recall, is a connective tissue. Um, Take a moment to see if you remember what the definition of a connective tissue is. If you are thinking that it is cells suspended in an extracellular matrix, then you're remembering properly from Anatomy and Physiology 1. So you've got your white blood cells and your red blood cells floating around in this matrix, which is plasma. The red blood cells we call erythrocytes, the white blood cells we call leukocytes. And then you have these things called platelets, which aren't cells in and of themselves. They're fragments of a larger cell, and that's why these three things are collectively called the formed elements rather than your blood cells, because the platelets are not cells. So when you take this connective tissue, you can separate out the cells from the extracellular matrix that they are suspended in. The cells settle out to the bottom because they are heavier than the plasma. The red blood cells, or erythrocytes, being the most dense, settle to the bottom with the white blood cells on top. So when you do this, you can calculate or measure what percentage of your column of whole blood is taken up by your erythrocytes. We call that the hematocrit, and it gives you an idea of what the oxygen carrying capacity of your bloodstream is. And sorry, I'm just going to delete that. Click to add title. There we go. So hematocrit is the measure of what percentage of your blood volume is taken up by your red blood cells. These are the average values for males and females. So in your average female, 42% of her bloodstream, or of her blood volume, is taken up by red blood cells. This is important because your red blood cells carry oxygen. So your hematocrit is a rough measure of the oxygen carrying capacity of your bloodstream. Going back to plasma, it is, as it says right there, mostly water, 90% water. There are a lot of proteins in your plasma. Many of these are produced by the liver, and we don't really have time to go into all the different kinds of proteins that you find in the plasma. There are also byproducts of metabolism, many of which contain nitrogen. The most common nitrogenous waste in your bloodstream is urea. And if you think back to chapter 3, try and envision which of your three macronutrients, protein, carbohydrates, or lipids, contained nitrogen. If you're thinking nitrogen is a component of the amino acids in your protein, then you're on the right source, or on the right track. So urea is what happens to the amino groups which contain the nitrogen on all of your amino acids if you don't convert them into proteins. So any amino acids that you ingest that don't end up incorporated into one of your body's proteins gets metabolized as part of your carbohydrate metabolism. They strip off the nitrogen containing amino group, or your cells strip off the nitrogen containing amino group, and the rest of the amino acid is metabolized for energy. The amino group becomes urea, and that gets excreted. So you've also got creatinine, which comes from your muscles. If you remember creatine phosphate, that's where the creatinine comes from. Nutrients we don't care about, or they're obvious. Electrolytes, you've got some electrolytes in your bloodstream. Hopefully you can recall what some of our key electrolytes are. I'll give you a second for that. So you've got sodium, potassium, calcium, probably the three major cation or positively charged electrolytes. And then these two are your more popular or more abundant negatively charged anionic electrolytes, your chlorine and your bicarbonate. You also have respiratory gases in your plasma. These are what you exhale, which is your CO2, and what you inhale, which is your oxygen. You have a lot more CO2 in your plasma than your oxygen. If you recall, we already said that the red blood cells carry the oxygen, and this is because it's not very soluble in water, so you don't find a lot of oxygen in your plasma, whereas CO2 dissolves readily in water, which is why soda and beer are fuzzy, champagne as well. Hormones, of course, we've already mentioned. All right, so now we're on to the formed elements. This is kind of the meat of the chapter, and we're going to be talking about white blood cells, red blood cells, which is all of these things here are your white blood cells, and then platelets. So you'll notice that they're just little tiny fragments. They're even smaller than your red blood cells. You might be noticing that the red blood cells don't have any dark purple blob in the middle. Right? They're missing the nucleus. So anything that's got a dark purple blob in the middle is some sort of white blood cell. And I don't know why the platelets stain so dark. They do not have any DNA in them, but they're probably just densely packed with proteins that like to absorb that dark stain. All right, so erythrocytes. These are your red blood cells. The color is biconcave, which just means it's concave on top, it's concave on the bottom. This gives them a very large surface area to volume ratio, so that an oxygen inside a red blood cell doesn't have to travel far to get out, and an oxygen outside of your red blood cell in the lungs doesn't have to travel very far to get in. So all of your hemoglobin molecules are about as close to the surface as they can get with this shape, which makes sucking up and spitting out oxygen easy. We already said they don't have a nucleus. They don't really have very many organelles. They're basically just these neatly shaped bags of hemoglobin that transport oxygen, and they are very well shaped or designed to do so. So we just mentioned hemoglobin. This is the molecule that actually carries the oxygen. This is what hemoglobin looks like, sort of diagrammatically. It actually contains 4 different peptide chains, which are globular in shape. That's where the name comes from. So 1, 2, 3, 4. If you recall from Anatomy and Physiology 1, if you have multiple peptides in the same protein, that's quaternary structure. So here's an example of a protein with quaternary structure. So each one of the peptides has a pigment in the middle, which is called heme. In the middle of the heme is an iron. It is the iron that can bind oxygen. So each heme has one iron. Each iron can bind oxygen. You have 4 hemes in a hemoglobin, so each molecule of hemoglobin can carry up to 4 molecules of oxygen. And then you want to remember that there's like billions of these inside every red blood cell. So a red blood cell can carry millions or billions of molecules of oxygen, but they're carried 4 at a time, max, on all of the hemoglobins. All right, just terms to know. Hemoglobin, when it's got oxygen on it, is called oxyhemoglobin. Easy enough. Hemoglobin, when it doesn't have oxygen on it, is called deoxyhemoglobin. And we are not going to worry about carbaminohemoglobin because we have enough stuff to worry about. And if you look here, the oxygenated hemoglobin is very red, brighter red, like a ruby color. And the deoxygenated is darker. This is because when iron binds oxygen, you get essentially rust, and rust is kind of red. You're oxidizing iron. This is how it gets carried in your bloodstream. So the oxidized hemoglobin is redder than the deoxygenated, or not oxidized hemoglobin. That's why your arteries are colored red and your veins are colored blue, because this darker red looks kind of blue through your skin. So before we talk about red blood cells specifically, we have to talk about blood cell formation. Or I should say, before we talk about how red blood cells are formed, which is coming up next, we will talk about blood cell formation in general, which is called hematopoiesis. It takes place in the bone marrow. You are going to see that some of your blood cells migrate to other parts of the body to complete their development, but they're born in the bone marrow. All of your red blood cells, white blood cells, and platelets ultimately come from the bone marrow. This is the red marrow that you find in the spongy bone of your bones. So the epiphysis of the humerus and the femur both have a lot of spongy bone, and other bones, I'm sure. So the way it works is that you have one stem cell called the hemocytoblast, and depending on which hormones or growth factors it's exposed to when it's going through mitosis, it will take on any one of the different developmental pathways that lead to all of your different formed elements. And this is one of the cell populations that people refer to when they talk about adult stem cells. So the hemocytoblast can keep going through mitosis after mitosis. One of its daughter cells will go on to complete one of these developmental pathways, and the other daughter cell will remain a hemocytoblast and go through mitosis again and keep replenishing your blood cells. So the production of red blood cells is a specific type of hematopoiesis called erythropoiesis. It is promoted by the hormone erythropoietin, which you probably remember from the first exam now is produced by the kidney. So when the kidney senses hypoxia, it produces EPO. EPO makes it into the bloodstream. When it gets to the marrow, the hemocytoblasts churn out more erythrocytes. If you're wondering what hypoxia is, just think about it for a second. Hype generally means below. You've kind of got hypo there. And then the O is also shared by the oxia, and that refers to oxygen. So if you've got hypoxia, you've got reduced oxygen. So if the kidney's not sensing oxygen, then it releases erythropoietin, and you make more red blood cells to increase the oxygen carrying capacity of your bloodstream. So as I just said, hypoxia is low oxygen levels in tissues. This could be caused by a number of different reasons. Kind of the most obvious reason is the second one listed here. It could just be that there isn't a lot of oxygen in the bloodstream. But it could also be that that tissue isn't getting a lot of blood. So if you have blockage of one of your coronary arteries, then you've got hypoxia in your myocardium or your heart muscle. So that would be an example of a lack of blood causing hypoxia as opposed to a lack of O2. And one of the reasons why you might have less oxygen in your bloodstream would be reduced availability of oxygen. And you're going to see later that we will talk about defects or problems with your red blood cells that would reduce their oxygen carrying capacity, which would be a lack of O2 which would lead to a lack of O2 in the blood. And we'll talk about that as well and make sure that we keep that separate from hypoxia. So your red blood cells don't live forever. None of your blood cells live forever, in fact. Your white blood cells do die off at a certain rate, as do your platelets. Your red blood cells, I think, sort of being one of the more numerous ones, and they tend to live the least amount of time because they don't have a nucleus, have to be continually removed from the bloodstream, and it is the job of the spleen to do that. Where's spleen? There it says spleen. So for us, that's really what we care about the spleen, is that it's got macrophages, which are phagocytes, that engulf your dying red blood cells. This next slide does contain the information on a possible open-ended essay question. And the question is just what happens to your red blood cells after they die, and how are the components of hemoglobin processed, I guess, for lack of a better word. I forget exactly how the question is worded. So as it says right here, the dead red blood cells are phagocytized in the spleen. The iron, because it is a mineral and you don't get tons of it in your diet, is stored generally in your liver and then somehow makes it back to the bone marrow so that it can be incorporated into new red blood cells. The heme itself is degraded. For whatever reason, your body decides it doesn't need to hang on to it. It has the energetic ability to just make more heme. So that gets turned into a bile pigment called bilirubin, and it is one of the reasons why your feces has the color that it does. And then the globin, if you remember, is a protein. So it gets broken down into its individual amino acids, and they are either processed for energy or recycled into other proteins. So this brings us back to oxygen and the discussion of anemia. So anemia is just, as it says here, low oxygen carrying capacity in the bloodstream, and it can be due to a number of different reasons, and it is one of the possible causes of hypoxia. So the first reason you might not be able to carry much oxygen in your bloodstream is that you just don't have enough red blood cells. Perhaps you have bled out, you've hemorrhaged, so you've lost some red blood cells. It might be that they're dying for some reason and busting open. Whenever a cell busts open, that's called lysis. So hemolytic anemia is red blood cells busting open anemia. And then aplastic anemia just means that your bone marrow isn't making enough red blood cells for one reason or another. Another reason that you might have low oxygen carrying capacity would just be that you don't have enough hemoglobin, perhaps because you don't have enough iron in your diet. The third cause of anemia is abnormalities in the hemoglobin itself. Generally a genetic abnormality that leads to the production of an abnormal hemoglobin. These are called thalassemias, and they affect not only the shape of the protein chain or the globin itself, but also the shape of the red blood cell. And I don't really understand how that works, I just know that it does. So one of the more common types of thalassemia is sickle cell anemia, which we have a picture of here. So again, these are genetic problems. You have a normal person, the sixth amino acid in their hemoglobin chain is glutamate or glutamic acid. If your DNA gets mutated, or I shouldn't say mutated, if you inherit a mutation such that your hemoglobin contains a valine there, a different amino acid. It changes the shape of the hemoglobin. That changes the shape of the cell from the biconcave disc up top to the sickle cell shape. That disrupts the surface area to volume ratio and also makes it harder for the hemoglobin to carry oxygen. So it decreases your oxygen carrying capacity. Then you have pernicious anemia, which I almost never talk about, but it's just a lack of vitamin B12 that they treat with a B12 injection. Right. And we are going to stop there. The Tuesday, Thursday section is a little bit further ahead than this, but I think there really are limits to how much we can cover with screencasts. So I will stop here. Thank you for bearing with me thus far. That's it.